Okay, so let's begin. Hello everyone, welcome to Environmental Research Webinar. So I am Sungkyun Park, a faculty member at Epidemiology and EHS. So we are very excited to host this exciting topic. And so we will introduce our speaker later. So this seminar is sponsored by Michigan Life Stage Environmental Exposure and Disease Center, in short, M-LEEaD. And I will introduce today's moderator, Dr. Xin Wang, who is a M-LEEaD Center Scientist and Research Assistant Professor in Epidemiology. So I will take it over to Dr. Wang.
So good afternoon, everyone, and welcome to today's M-LEEaD seminar series. And it is my great pleasure to introduce our speaker today, Dr. Donghai Liang. And Dr. Liang is an assistant professor at Emory University Rollins School of Public Health with primary appointment in the environmental health and a secondary appointment in epidemiology. And his research is at the forefront exposure science and molecular epidemiology, where he has contributed significantly over 70 peer-reviewed papers. His work in air pollution assessment, health research, and omics-based technologies has been recognized widely. And as a faculty member of the Emory Hercules Exposomics Research Center, his research has been instrumental in measuring the multidimensional exposures and elucidating the molecular mechanisms that account for the complex health responses to the environmental mixtures, including air pollution and persistent organic pollutants. More recently, Dr. Liang has shifted the majority of his efforts towards incorporating the high-throughput omics technologies, especially high-resolution metabolomics, into investigations on the molecular mechanisms and the disease etiology associated with environmental exposures. Today, Dr. Liang will share his insights on the use of high-resolution metabolomics to measure the internal exposures and investigate the molecular mechanisms accounting for the complex health responses to ubiquitous environmental pollutants. So please join me in welcoming Dr. Liang.
Thank you very much. Hello, everyone. My name is Donghai Liang, and I'm an assistant professor of Emory University. Before I start my talk, just want to check if you can see my screen well.
Yeah, we can see the screen.
Okay, perfect. Thank you very much. And again, thank you so much for the nice introduction, Dr. Wang. So today I'm very happy to be here and talk about the recent work that we do in the environmental metabolomics and exposomics research group at Emory, the Emerge group. And I'm excited to present our recent work in applying high-throughput omics technology, especially high-resolution metabolomics in exploring the mechanisms underlying the toxicity of environmental pollutants on maternal and child health and among vulnerable populations. So the motivation for us to conduct this research really lies in the series public health threat caused by environmental exposures. Take air pollution, for example. As you can see here, according to the recent statistic from the Global Burden of Disease study, air pollution is the highest ranking environmental risk factor for mortality, with more than 6 million people die, unfortunately, prematurely due to exposure to household and ambient air pollution every year. So despite the well-documented epidemiological evidence linking numerous adverse health effects shown here that associate with both short and long-term exposure to air pollution, the detailed molecular mechanisms and pathways underlying the toxicity of air pollution on human health remain largely unknown due to the lack of sensitive and specific biomarkers to assess the internal exposure to air pollution. So thanks to the development of high-throughput sequencing and analytical technology, many omics-based approach shown here have emerged as promising analytical platform that can potentially tackle these challenges. So for my talk today, I will mainly focus on the metabolome, which is the large-scale global study of small molecules commonly known as metabolites in the body. So as you can see here, compared to other omics layer, the metabolome is the final downstream product of the gene transcription and the gene environment interactions. And therefore, the changes in the metabolome are amplified relative to the changes in the genome, epigenome, transcriptome, and the proteome. And additionally, as the downstream product, the metabolome is closest to the phenotype of the biological system. At Emory Hercules Exposome Research Center, under the leadership of Dr. Dean Jones, we have developed an efficient, high-resolution metabolomics platform that's capable of detecting and characterizing tens of thousands of metabolic features from each biosamples. And as a consequence, compared to the traditional targeted analytical approach, just focusing on a limited number of less sensitive biomarkers, high-resolution metabolomics might offer a more sensitive measure of internal exposure and generate novel hypotheses to understanding the toxicity of environmental chemicals on human health. So before I show you how we apply this platform in our own study, I would like to first briefly go over this untargeted metabolomics workflow with you. So the first step is to conduct biomonitoring and collecting biosamples from our participants in the field. And there are many biospecimens, as you can see here, that are suitable for metabolomics application, including blood-based measure, saliva, exhaled breath condensate, urine, and fecal samples. Then these samples will be analyzing using liquid chromatography or gas chromatography coupled with high-resolution mass spectrometries. So at Emory, we mainly use LC-MS using multiple chromatography column, including the helic hydrophilic column, as well as the C18 hydrophobic column to enhance the coverage of the feature detections. So after the experiment, we will use two advanced in-house developed algorithms to extract metabolic signals from this raw data generated from LC-MS. And the final product is a feature table which contains key information for three major characteristics for each of the metabolic features, which include the mass to charge ratio, which is the molecular mass of the cation divided by its charge, the retention time, which reflects the time taken for each of the metabolic features to pass through a chromatography column. And finally, we also have intensity detective, which is the signal strength of the peak for each of the metabolic features, which can serve as a surrogate of their relative concentration in the biosamples. So once the feature table is generated, we will integrate this data with external exposure profile we detected, along with the health outcome data we measure. Then we construct statistical model to conduct this metabolome-wide association study, the M-LEEaD statistical modeling, to investigate what metabolic signals are associated with the exposure or the outcome of interest. After running these M-LEEaD models, we usually will identify tens or even hundreds of significant features after false positive discovery rate corrections. However, we know nothing about their chemical identities. So to predict the biological function of these unknown features, we'll conduct pathway enrichment analysis using several advanced bioinformatic tools. And finally, based on the pathway enrichment analysis results, we will conduct chemical annotation on those features that are not only associated with exposure, but also enriched in a pathway that are relevant to our research interest. Then we try to compare the mass to charge ratio, retention time, and the extracted ion chromatogram to the authentic reference standards to confirm their identity with level 1 evidence. Finally, we will look at this confirmed metabolite and see if this have established any dose-response relationship with exposure or the health outcome indicator, and further explore the underlying mechanisms by generating new hypotheses to explain how these metabolites are linked with the exogenous pollutant and how they contribute to the observed health responses. So using this powerful platform, my team has successfully assessed the internal exposure and mechanisms underlying the toxicity of complex environmental exposure on different health outcomes in more than 12 independent cohorts. And today I'm going to highlight some of this study and show you how we use high-resolution metabolomics in environmental research. So the first study I want to introduce here is called the dorm room inhalation to vehicle emission to drive study. This is also one of the very first US study that used high-resolution metabolomics in air pollution health research. So our study location, if you can see here, is at the center of the metropolitan area of Atlanta. If you look at the yellow dashed line, this is the I-75 and I-85 highways connector, where there are more than 300,000 vehicles passing by on on a daily basis before COVID. So we selected 2 dormitory locations where our Georgia Tech student will live in. One is being the near dorm, which is only 20 meters away from the highway, and the other is the far dorm, which is 1.4 kilometers away from the highway. And our hypothesis is that the student participants who live in these 2 different dormitory locations will be exposed to differential level of traffic-related air pollution exposures, and that these differences can be further detected by looking in their metabolomics profiles. And also during this 4-month sampling period, we conducted extensive exposure assessment using continuous sampler, integrated sampler, as well as local sensors to characterize individual level exposure to traffic-related air pollutions. At the same time, we recruited 54 college students who just moved in these 2 dormitory locations, each of which we give them GPS monitor to record their location, how much time they spent, and how far they were away from the highway. And knowing that a lot of other factors will impact human metabolomics profiles, we also asked each participant to fill in the questionnaire to record their dietary pattern and health status. For the biomonitoring, we collected longitudinal saliva and blood samples for high-resolution metabolomics profiling, which I just show you guys a little bit using the workflow that we developed at Emory. So here shows you the results for the traffic exposure assessment. As expected, for most of the participants who live in the near dormitory locations in green, they were exposed to a higher level to this traditional traffic pollutant indicator, including carbon monoxide, nitric oxide, and carbon, compared to the participants who live in the far dorm locations. Then the key questions become whether or not we can detect the internal biological signals that can reflect this exposure contrast using high-resolution metabolomics. So after running more than 1 million metabolome association study modeling across different biometrics using different traditional traffic-related air pollutant indicators, we found out that there were hundreds of metabolic signatures that are significantly associated with at least one or more of the traffic pollutant indicators after covariance adjustment and after adjusting for false positive discovery rate. We then conducted pathway enrichment analysis and chemical annotation to see if we can identify some of these promising significant features. So here is a heat map summarizing the metabolic pathway that are associated with at least 3 or more of the traffic-related pollutant indicators. Each of these tiny cells represent the association between each biological pathway and each of these traffic pollutant indicators. And the color was shaded according to the p-value of the associations. So in plasma samples, as you can see here, there are 24 pathways consistently showing strong association with traffic pollution. And 10 pathways highlighted here have been previously reported to be involved in oxidative stress and inflammation processes induced by air pollution exposure in the animal model or in cell line model. And more excitingly, when we look at the heat map for the saliva sample using the same chromatography column, we observe very similar suite of pathway popping up, especially the leukotriene metabolism as well as the vitamin E metabolism, which were consistently the top 2 pathways captured in both plasma sample and saliva samples. When compared to the reference standard, we were able to confirm the identity of 9 significant metabolites with level 1 evidence. All of these confirmed metabolites you can see here are closely related to oxidative stress, inflammation, and DNA damage and repair mechanisms. Specifically, chlorine, arginine, histidine, and hypoxanthine have been previously used as a pulmonary inflammation indicator in the in vivo models, and they were also associated with nitrogen oxides and PM modifying. Consistently, we observed similar results in our own study. So in this proof of concept project, we demonstrated that high-resolution metabolomics was capable of detecting sensitive and robust metabolic signals and pathways that closely linked to traffic-related air pollution. Although we're very excited about this result, the generalizability and the consistency of these results need to be tested in other independent cohort. So to further test our findings, we conducted the same analysis, but this time in another cohort called the Atlanta Commuter Exposure Study. Here, we focus on participants with and without asthma, and we also want to see whether or not the metabolic associations with traffic-related air pollution will differ among participants with or without asthma. So here are some brief background on this study design. So during the study period, our study participants will come to our lab in about 7 o'clock in the morning. Then they will conduct a scripted 2-hour commute on one of the most congested highway in Atlanta. After 7 days, they will participate in another exposure scenarios where they were randomized to either sit in a clean indoor environment without any traffic exposures, or they will do another additional commute, but this time on a side street, a surface street with less traffic volume. And within the vehicles, as you can see here, we have put tons of instruments to measure their personal commute exposure to different traffic-related air pollutants, including 27 different indicators of traffic exposures. And a total of 24 participants with asthma and 21 without asthma contributed their blood samples before and right after each of the exposure scenarios for the metabolomics profile. Here shows you some exposure contrast we observed in different exposure scenarios. And again, as expected, for most of these traffic-related air pollutants shown here, their exposure level was significantly much higher during the highway commute scenario compared to the surface street commute or the clinic environment. Then we'll use high-resolution metabolomics to identify biological signals that can reflect this exposure contrast. So here is a Venn diagram summarizing our pathway enrichment analysis results. So first of all, there were 9 metabolic pathways consistently showing strong association with traffic exposure, and they're all shared by all of the participants regarding whether or not they have asthma. As you can see here, this includes inflammation and oxidative stress-related pathways such as leukotriene metabolism, vitamin E metabolism, as well as CYP450 pathways. And again, this is also consistent with our previous findings in the DRIVE study. And more interestingly, when we stratify the panel by asthmatic status, we observed that among those participants who have asthma, their unique top pathway that are closely related to acute inflammation responses, including the arginine and taurine, methionine, cysteine, and tyrosine metabolism, that this pathway only shows up among the participants with asthma. Then using the chemical reference data, we are able to confirm 45 metabolites with level 1 evidence. And more interestingly, 32 of these confirmed metabolites, with some of them shown here, were exclusively found to have differential association with traffic exposure between participants with and without asthma. And take plasma arginine level, for example. Arginine is an amino acid that involved in anti-inflammation regulations. And in our analysis, as you can see here, for most of the participants with asthma, when they're exposed to higher level of traffic exposure, their arginine level actually decreases. And only contrary, when you look at the healthy participant without asthma, their arginine intensity actually increases when they're exposed to higher level of traffic exposure. In addition to arginine, we also observe a similar pattern for pulmonary inflammation-related amino acids, including histidine and methionine. Based on this non-metabolic network, we found out that most of these validated metabolites were closely connected endogenously, centering around the arginine metabolism. Then our question becomes, what exactly is the role of traffic exposure in triggering the perturbation of the arginine metabolome? And how does asthma status further modify these metabolic responses? So by looking in the literature, we came up with this hypothesis. First, increased traffic exposure might induce the augmentation of arginase. And as reported in the previous studies, asthmatic status can further amplify this effect by reducing the bioavailability of arginine, which will trigger the downstream airway dysfunctions. And consistently in our own analysis, we observed that following the increase in traffic exposure, there's a decreased intensity in arginine as well as its precursors, including citrulline, glutamate, cysteine, and choline, and 5-oxychlorine. At the same time, we also observe increased level of chlorine as a result, which will finally contribute to the downstream adverse health impact on the respiratory system. There are also several other potential mechanisms, but that due to time being, I won't go into details. Throughout these processes, we can see how powerful high-resolution metabolomics application is by enabling us to really look beyond the line of the lamppost. So in traditional targeted analysis, we are usually limited to looking at a very narrow part of the molecular network biological responses given to the limited number of biomarkers that we can use. With metabolomics capacity of measuring tens of thousands of features, we might be able to see a much larger pathway and system-wide changes that can potentially link to each other and connect these molecular puzzles together. And as a result, it might provide a more comprehensive and accurate biological picture of what's really happening when our body are exposed to higher level of air pollutions, something that we might not be able to use by just using the targeted approaches. So in addition to the DRIVE and the ACE study, we also successfully applied this powerful platform in several other independent studies among healthy adults, participants without asthma, women who underwent assisted reproductions, in panel study, crossover trial, and in prospective lung cancer prevention studies. And more recently, we have focused on underserved and underrepresented populations among African-American and Hispanic pregnant people and children. So here we focus on preterm birth and early term birth, which are significant public health problems. Both preterm birth and early term birth have been linked with increased risk of adverse children and adult health outcomes in later life. In the past decades, if you look at the figure on the right, the prevalence of preterm birth and early term birth has increased gradually in the US. And unfortunately, this public health burden is not shared equally across the population, with Black and African American people persistently experiences a 50% higher rate of preterm birth and early term birth compared to other races and ethnicity group. However, most of the current air pollution epidemiology studies do not specifically focus on these marginalized populations who are disproportionately exposed to higher PM2.5 exposure and other air pollution exposure, and also have higher risk of adverse birth outcomes. Additionally, although previous literature has suggested inflammation, oxidative stress, endocrine disruption, and placental DNA methylation might be the potential mechanisms explaining the health impact of PM2.5 on these adverse birth outcomes, the detailed mechanisms and the molecules that are involved in this process is still remaining unknown. So to address this knowledge gaps, we got a R21 grant from EHS 3 years ago, and then we designed and conduct a traffic exposure, maternal, metabolome, and birth outcomes of tempo study to further use high-resolution metabolomics to understanding the mechanisms underlying the toxicity of traffic exposure on birth outcomes among 330 African-American mother and newborns. So here, my PhD student who recently graduated, Dr. Zhenjiang Li, for the first time adopted a parallel strategy using both the meet-in-the-middle approach as well as the high-dimensional mediation analysis to identify and validate mediated metabolic signatures that are indicative of the effects of PM2.5 exposure on both preterm birth outcomes and early-term birth outcomes. And our findings shows that there are about 10 metabolites shown here that were able to confirm with relevant evidence that are associated with both exposure to PM2.5 during different exposure time window of the pregnancy, as well as increased risk of early term birth and preterm birth. And coupled with the pathway enrichment analysis, we hypothesized this potential molecular network that are centered around the metabolism of thyroxine, urea cycle, proline, as well as leucine, to be potentially the overlapping molecules that may be further explain the effect of prenatal exposure to PM2.5 on increased risk of preterm and early term birth. And many of these pathway and metabolites were also reported previously in our other individual air pollution metabolomic study. And as for the future direction, we have a pending NIH application to expand our work in this study by conducting multi-omics analysis, by not only just focusing on metabolomics, but also integrating other omics layers, including the genome-wide DNA methylation, the epigenomics data, as these omics layers are really closely connected with each other. And by focusing on multi-omics, this might provide us with a more comprehensive view on the mechanisms underlying the effect of air pollution exposure on adverse birth outcomes. And here I just want to quickly share some of the exciting preliminary findings we got from a subset of 100 participants, where we identified several clusters of interconnected hyper or hypomethylated CpG sites and metabolites that are associated with both exposure to air pollution during pregnancy, as well as associated with increased risk of preterm birth. And hopefully we will get this R1 funded soon to expand our analysis to the entire cohort of 700 participants. So not only do we observe consistent and robust results in our own individual studies, similar findings were also observed by other groups around the globe. So in the past 4 years, we have been working closely with colleagues from the European Exposomics Consortium, and we just published this systematic review where we summarize the result across all the existing 47 air pollution metabolomic study. And we were very excited to report that despite the heterogeneity in the analytical platform and statistical approaches, there are more than 50 metabolites and pathways shown here that are consistently reported to be associated with both short-term and long-term exposure to air pollutions. And we might be able to further develop some of these metabolic markers as sensitive biomarkers to assess internal exposure to air pollution upon future validation study. And for the remaining time of my talk today, I also want to highlight some of our other metabolomics investigation in emerging environmental chemicals, such as per and polyfluoroalkyl substance PFAS, which is a group of widely used long-lasting chemicals, and they break down very slowly over time. That's why we call them forever chemicals. So in the past decades, many epidemiological studies, including those amazing studies conducted by an investigator in the M-LEEaD Center, have demonstrated that exposure to PFAS can lead to adverse health effects, including negative birth and children's health outcomes. And despite these evidences, the underlying mechanisms linking PFAS exposure and adverse birth outcomes are complex and largely unknown, thus limiting the development of effective interventions. So to address these knowledge gaps, thanks to the support from the ECHO consortiums, I received the ECHO OIF funds and decided to conduct a PFAS exposure and child health study in the Atlanta African American maternal and child cohort. Here, our hypothesis is that elevated exposure to PFAS during pregnancy will result in perturbation in specific metabolic pathway, especially those linked to oxidative stress, endocrine disruption, and inflammation, which will then in turn increase the risk for adverse birth outcomes, including preterm birth and small for gestational age. And 2 years ago, we published this first paper from the PITCH study on Environment International, which is led by our recent PhD graduate, Dr. Zheyong Zhang. So here we focus on the early pregnancy samples that we collected from 313 African American pregnant people, and we then sent out this maternal serum sample during early pregnancy for both the targeted exposure assessment on PFAS as well as the untargeted high-resolution metabolomics profiling. And in this study, we observed significant association between maternal serum PFOA and PFNA concentration with reduced fetal growth. And this effect of the PFAS on fetal growth were also similar compared to what was already previously observed in other populations, as well in other ECHO-Y analysis. And at the same time, we also seen an increased risk of small for gestational age that are associated with increased level of PFOA and PFNA during pregnancy. Meanwhile, using metabolomics analysis, our pathway enrichment analysis indicating that the underlying biological mechanisms of the PFAS fetal growth relationship might be centered around these amino acids fatty acids, bile acids, as well as some energy metabolisms that are associated with both increased level of PFAS during pregnancy and also negative adverse birth outcomes, including low birth weight and small for gestational age. Moreover, we are able to confirm 10 metabolites with level 1 evidence that are associated with both the PFAS exposure and indicator of fetal growth. Specifically, uric acids, as you can see in our top row, which is identified as a potential intermediate metabolic signals that are associated with both exposure and the adverse birth outcome. Specifically, as you can see here, uric acid intensity was positively associated with increased level of both PFOA and PFNA during the pregnancy. And at the same time, when we see that with the increase in the uric acid resulting from the increased level of PFOA and PFNA, the birth weight, it will result in a negative association with the birth weight, meaning that when the uric acid goes up, the birth weight actually goes down among the baby that were born from the pregnant people. So in addition to uric acid, we also identify several others potential intermediate markers that can underlie the toxicity of PFAS on fetal growth. So this really centering around the perturbation of amino acids, lipid and fatty acids, bile acids, as well as androgenic hormones. So to quickly summarize our findings in this study, we found out that some PFAS exposure contribute to health disparity of reduced fetal growth in our African-American maternal child cohort in Atlanta. And then the potential underlying mechanisms might be centered around metabolic perturbation in uric acids, amino acids, lipid fatty acids, bile acid, as well as sex hormone metabolisms, which can further affect fetal growth. And to further validate our findings, we not only focus on characterizing the maternal metabolome, but we've conducted a follow-up study where we focus on this metabolic perturbation in the newborns. And just a few months ago, this analysis led by our PhD student, Caitlin Tybalt, was published on Nature Communication, which we look at the molecular signatures that are associated with both PFAS exposure and gestational age and birth outcomes in the newborn dry blood spot metabolome. And by focusing on newborn metabolome using the same high-resolution metabolomics workflow, we are able to identify over 600 overlapping metabolic signatures that are associated with both prenatal exposure to PFAS and increased risk of adverse birth outcomes. And comparing to the chemical standard, we are able to confirm a total of 56 overlapping metabolites with high confidence ontology levels. So this metabolite can be further broken down into 7 major categories, and I apologize for the relatively small font size shown here, but the take-home message is that the major metabolic perturbations are centered around the amino acids and proteins, as well as the chemical messengers, which are all shown to be associated with all of these 4 different PFAS species, but also at the same time increased risk for small for gestational age, preterm birth, and low birth weight. Simultaneously, we also identify 14 unique overlapping pathways that are associated with both PFAS exposure and gestational age and birth outcomes, with the top 2 metabolic pathways consistently showing the strongest association, including the metabolism of amino acids in the uric cycle and the tryptophan metabolism. And for the uric cycle, not only did we observe in the newborn metabolome, we also observed a similar pattern that shows up in their mothers during the pregnancies. So taking all these results together, we propose this potential molecular network that can potentially underline the toxicity of prenatal PFAS exposure on reduced length of gestation, which center mostly around tissue neurogenesis, neuroendocrine functions, and redox homeostasis. And again, although we are very excited about these results, we need to keep in mind that this is a hypothesis generation approach, and all of this metabolic network that proposed here needs to be validated using in vivo and in vitro models. And here is another very exciting project that I want to showcase here. So in traditional metabolomics study, we usually focus on the single pollutant one at a time. But in the real world, we are exposed to most of these pollutants simultaneously. So instead of studying each of the PFAS species individually, we have started using the advanced environmental mixture analysis, specifically the quantile G computations. And so this is the first time we use environmental mixture analysis in metabolomics study. And we observe metabolic perturbation in several consistent information of cities stress related metabolism that are associated with both the single PFAS indicator as well as the PFAS mixture. And here, when you look at the results when we use the mixture analysis, you will find out that when we consider all of these PFAS species simultaneously, we are actually able to detect a stronger association between the metabolic perturbations and the PFAS exposure, which indicated that in the future, in all of these environmental metabolomic studies, we also need to consider environmental mixtures as we are simultaneously exposed to all of them. And when we observe so many metabolic pathways, we kept wondering whether or not some of these pathways are specific to PFAS or air pollution exposure, or they will also show up in other pathways. So using the same high-resolution metabolomics analysis, we also investigate other emerging chemicals, including exposure to tobacco smoke, where we observe quite a unique metabolic network and pathway that are different from the PFAS or air pollution exposure. And here we observe that the tobacco smoke during pregnancy are associated with the metabolic pathway that's entering glycine glucose TCA cycle, which then further contribute to increased risk of adverse birth outcomes. We also conduct a similar analysis on other emerging chemicals, including phthalates exposures, bisphenol A, bisphenol F among African American during pregnancies. We also look at pesticides and insecticides exposure during the early pregnancy and see how they contribute to perturbation in human metabolome. We also reported our most recent work, which is focused on the metabolic associations with exposure to PBB in the Michigan PBB registry. And here, using high-resolution metabolomics, we are able to detect a lot of metabolic pathways and metabolites that are associated with both PBB and PCB. And our next step is really to look at how this metabolic perturbation will result in the observed health risks that are associated with PBB exposure in this cohort. And last but not least, we also started to use high-resolution metabolomics to look at the impact of other environmental exposure, including a heat exposure, and see whether or not they have a link with perturbation in human metabolome, which would then increase risk of acute kidney diseases among farm workers who works in the agricultural setting. So with that, I just want to conclude my presentation by just quickly summarize what we have learned so far and our future direction. So hopefully today I show you that metabolomics can serve as a sensitive platform that may link environmental exposure to reflecting internal doses and biological responses. Throughout all this application, we observe that environmental pollutant might induce perturbation in human metabolome, and this metabolic perturbation might then in turn increase risk of the adverse health outcomes. Because of the hypothesis generation natures of all of these environmental metabolomics application, the exact mechanisms by which environmental exposure impact human health still needs to be validated using hypothesis-driven protocol using in vivo and in vitro models. In the future, the application of multi-omics and mixture analysis are really critical for us to have a more comprehensive understanding of the molecular mechanisms underlying the toxicity of different environmental chemicals on human health. So with that, I want to conclude my study by really acknowledging the great funding support from the Emory Hercules Exposome Research Center, from NIEHS, from EPA, and the Health Effect Institute. I also want to thank my collaborators within Emory and outside of Emory for their consistent support and for providing access to different cohorts for us to really use this environmental metabolomics. And last but not least, I want to thank all my students and trainees trainees in the environmental metabolomics and exposomics research group for their great efforts in really leading this project. So with that, thank you again for inviting me to present in the M-LEEaD webinar. I'm happy to take any questions.
Okay, thank you very much, Dr. Liang, for that very insightful presentation. And then now we have about 10 minutes for the questions. And I see there are several questions coming in the Q&A box. So the first one is about what software or program are you using to conduct the enrichment analysis?
Yeah, thank you so much for the great question. So let me see if I can go back to my previous slides very quickly. Mainly, we use two in-house developed algorithms. One is called Multi-omics, and the other is called Metabolic. So both of these pathway enrichment analysis tools really aim at to incorporate the our existing knowledge of the non-metabolic pathway network, and then trying to predict the biological function of these unknown metabolic signatures without identifying them first. So by doing this pathway enrichment analysis, we will have a chance to understanding the underlying biological function, and then this will also help accelerate our processes of chemical annotation by just focusing on those metabolites that are involved in the most relevant pathways. And I'm happy to send you the link to both the software, but I think in this slide, you should be able to find out the access to both of these software.
Thank you for the answer. And the second question is about the Timbo study you showed in your presentation. So in that study, did you only use the maternal blood, or are there any other samples included in your analysis, like the cord blood and some other matrix?
Yeah, that's a great question. So as I showed previously, there are many different biospeciesmen that are suitable for metabolomics profiling. So in the Atlanta African American maternal and child cohort, in addition to the blood samples, we also have access to other different biospeciesmen, including dry blood spots, as well as urine sample and fecal sample collected from the children. So we don't have existing data yet, but hopefully with some of our future grants get funded, we will be able to also look at, like, for example, fecal samples and see if the metabolic signature that in the fecal sample can also somehow reflect the metabolic signature we observe in the blood. Because as we all know, for maternal and child health study, blood collection is relatively very invasive. So we have been trying our best to see if there's other non-invasive biometrics that we can use in understanding the impact of environmental chemicals on human metabolite in relation to health diseases. So in the DRIVE study, we show that both the saliva samples and the blood samples have really common and overlapping metabolic signals that shows up there to be associated with traffic-related air pollution exposure. So other interests of my research group is really to see if in the future we can maybe use saliva or fecal sample as more like a non-invasive sample for environmental metabolomics investigation, especially in maternal and child health.
Thank you. And the next question is that in the several your study presented today, you consider multi-hypotheses simultaneously, either on the multiple chemicals or the omics biomarkers or the outcomes. Mm-hmm. So which significant level was taken to declare the statistical significance? And did you consider any ways to adjust for the multiple comparison in those testings?
Yes, that's a great question. So one of the major limitations of all of this, like, high throughput data analysis is really the increased risk of the type 1 error, because we are repeating the same hypothesis, the same statistical testing on thousands of different chemicals collected from the same individuals. So we have, in all of the study that we present today, most of them have applied the most stringent FDR corrections at the FDR less than 0.05 using the Benjamin Holzberg procedures. Having that said, in some of the other initial cross-sectional study that we use, which we conducted as more like a hypothesis generation approach. Because of the relatively small sample size in some of those studies where we only have less than 100 people, we might not be able to conduct the same rigorous FDR correction. So we sometimes will have loosened our FDR FDR threshold to FDR less than 0.2, or sometimes you even just use the low p-value less than 0.05. This was done because some of this pathway enrichment analysis, they require at least 50 to 100 significant features as the input for their simulation. So we have to again balance the risk of type 1 and type 2 error. But for most of the time, we will, at least for the chemical annotation, we will only report those metabolites that survive the FDR correction, because we really think that we need to control for this type 1 error.
And there are more questions now flooding in. And there's one speaker's question asked, what does the overlap in the table of QG computation mean?
Yeah, sorry for this roller coaster showing of the slide. But then for the QG-com analysis, the overlapping is really showing you that the different metabolic pathway that are associated with individual PFAS and also the PFAS mixture. So what we found out here is that, as you can see here, when we conduct the environmental metabolomics study on individual PFAS species, we see some of this metabolic pathway pop up, but they're not really showing a consistent pattern across these different PFAS species. But when you consider this PFAS mixture using this mixture analysis simultaneously, you will see that a more consistent pattern. And then when we use the mixture analysis, we are able to not only just detect those pathways that shows up that are associated with single indicator, but sometimes we were able to detect novel metabolic pathway that do not get detected in other single PFAS, which may give you a more comprehensive idea of the metabolic perturbation associated with simultaneous exposure to PFAS.
Okay, we have 3 minutes left, and there are 2 related questions. So the first one is, could you please share if your group has used the MS2 annotation for those studies? And the second one is, could you please share if the MS1 annotation database, share the MS1 annotation database of your choice that provides annotations at a relatively high confidence?
Yeah, that's a great question. So for most of the confirmed metabolites that I shown here today, and also in our papers, these are all done using and all confirmed using the chemical reference standard that were validated through tandem mass spectrometry. And we have built up an internal library of more than 700 metabolites that we have used the same analytical approach using a tandem mass MS to confirm the identity. So most of these metabolites I report today, if I say that they have been confirmed with MS1 evidence, these are the ones that have been validated using tandem mass spectrometry. For the MS2 annotation, we have some of them, but usually we just provide the list of this MS2 annotation in the supplements, because we don't have the internal library that we are not 100% confident to really ensure about their chemical identity. And sometimes we also reported level 4 or level 5 annotations, but these are mostly done by just compared to the mass to charge ratio to the existing known database. But we are well aware that for the same mass to charge ratio, they can correspond into tens or even hundreds of different chemicals. So we usually do not report those metabolites that are confirmed with MS evidence that are lower than level 2.
Okay, I think we have time for the one last question. So Samkyun, do you have anything to ask here?
Yes, yes. This is a great presentation, and you did a lot of this, the metabolome study in across different population, different age group, different outcome, which are fantastic. So we really appreciate your this comprehensive analysis and result. And one interesting finding I found was you look at the saliva and blood in in the first study you show, and so the choice of this different biospecimen is not just because of the a convenience or invasiveness, but more likely whether the long-term effect or short-term effect, or this metabolite or effect of this chemical can be circulated in specific target organ and then this biospecimen. So can you give us a little bit more about the choice of this saliva and then blood and in this metabolomics study? So for the investigator who will be using this metabolomics in their future studies, so what should be considered to choose this different biospecimen?
Yeah. Thank you so much for the great question, Dr. Park. So you're right that for the saliva and blood, when we decided to measure both, it's not just because saliva is non-invasive. And on the other hand, we also want to see really whether or not saliva itself can reflect some sort of this short-term exposure to air pollution, and whether or not the signals are similar across blood. So the conclusion we saw from the DRIVE study is that in terms of the acute effect of air pollution, we do see there's some similar signals that are associated with metabolic signatures that in both saliva and blood. However, when we look at more a long-term effect, I didn't show it here, but then in terms of the metabolic signal that are associated with long-term exposure to traffic exposure, we saw much fewer signals in the saliva, which indicated that I think I hypothesized that saliva is still quite different from blood in terms of it also have the impact of oral microbiome and other external exogenous chemicals there. So saliva, in my opinion, might be a better biometrics to capture acute or short-term effect to air pollution exposures. But having that said, if we are able to conduct this analysis again or expand our sample size a little bit more, then we will be able to really look at both short and long-term exposure to air pollution and see whether or not we can also capture some chronic effect of environmental exposures on saliva and metabolome.
Great. Yeah, it's interesting to see that. So you see some consistent finding between saliva and blood for a short-term exposure, but more inconsistency with the long-term exposure. So yeah, I think it is very important message to the future investigator who will be using this platform. Hey, so we have no more questions? All right, so then, thank you, Dr. Liang, for this great presentation, and also Dr. Wang for facilitating this Q&A session. And for everyone who attended this seminar, so I hope you have a great Thanksgiving holiday, and we will see you again after the break. Thank you very much.
Thank you. Happy Thanksgiving to everyone.